Hey, welcome to the Explainer. All right, today we are diving deep into molecular cloning. This is really the engine behind so much of modern genetic engineering. And by the end of this, you're going to have a really solid grasp on exactly how it works. So let's get right into it.
Okay, so picture this. You're in a library, a massive one with a million encyclopedias, and your job is to find one specific sentence and copy it. Sounds impossible, right? Well, that's pretty much the challenge scientists face with DNA. An organism's entire genome is just unimaginably vast, but often they only need to study one tiny little piece of it, a single gene. And molecular cloning, that's their incredibly clever solution to this massive copy-paste problem.
So, first things first, let's just take a second to really wrap our heads around the scale of this puzzle. We're not just looking for a page in a book. Nope, we're looking for a single sentence on that page, inside a library that has millions of books.
You can think of a genome as the complete, unabridged instruction manual for an organism. And every single cell has this entire library. But what if you only need the instructions for building, say, one specific protein? Well, that specific instruction is your gene. So what cloning is all about is finding that one single page, pulling it out, and then making millions and millions of photocopies so you can finally read it clearly. And making copies, that's something nature is just ridiculously good at. So this number you see here, over 2,000, what's that about? Well, it gets at just how complex this is. To copy its DNA, a simple little bacterium has just one starting point. But our human cells, we have over 2,000 of these origins of replication, all working at the same time. It's this incredible natural machinery that scientists have figured out how to hijack for their own purposes. Okay, so to actually go in and manipulate DNA, you're going to need the right tools. So let's open up the scientist's toolkit that makes all of this possible. It's really all about precision cutting and pasting, just on a scale you can't even see. And really, it boils down to two essential tools: restriction enzymes and DNA ligase. The easiest way to think about them is as a pair of molecular scissors and a tube of molecular glue. The scissors cut the DNA, but only at very specific places. And the glue, well, it joins the pieces back together where you want them. They are the absolute foundation of this kind of genetic surgery. Now, with these scissors, the name of the game is precision, absolute precision. That's why scientists use what are called type 2 restriction enzymes. These things are incredible. They don't just snip DNA randomly. They scan along the DNA looking for a very specific sequence of letters, often a palindrome, which is a sequence that reads the same forwards and backwards. And they cut right there and only there. And this table, it shows you exactly what I mean by precision. Just look at these two examples, EcoRI and Hind3. Each one has its own unique code that it's looking for. You can see right here that EcoRI will only ever cut at the sequence GAATTC. This unbelievable specificity is what allows scientists to carve their gene of interest out of a huge genome with perfectly predictable clean edges.
All right, so you've used your scissors, you've cut your gene out. What now? You can't just let it float around in a test tube. You need some way to carry it, to protect it, and to get it where it needs to go. You need a delivery truck.
Well, that delivery vehicle is called a cloning vector. It's usually a small circular piece of DNA called a plasmid, which scientists first discovered in bacteria. Seriously, the best way to think about it is like a biological USB drive. You can insert your gene, your data file, right into the plasmid, and that plasmid will carry it safely into a host cell.
So for a vector to actually work, it needs three essential things. First up, an origin of replication. This is basically the start copying here signal, so the host cell knows what to do with it. Next, it needs a selectable marker, which is usually an antibiotic resistance gene. This is a super clever trick we'll talk about in a minute that helps us find the right cells. And third, it needs unique restriction sites. Think of this as a predefined docking station where we can paste our gene. Okay, so we've got our gene loaded onto its delivery truck. Now we need to send it somewhere. We need a factory that's going to mass produce it for us, making millions and millions of copies.
And for this job, the absolute workhorse is usually a totally harmless, lab-friendly version of the bacterium E. coli. What scientists do is introduce that vector, our USB drive, into these bacteria. And then the bacteria do all the heavy lifting. As the E. coli cells grow and divide, they also copy the plasmid and our gene right along with their own DNA. They become tiny living photocopiers.
So why E. coli? Why this specific bacterium?
Well, it's pretty much perfect for the job. It's super cheap and easy to grow in the lab. It replicates incredibly fast. We're talking it can double its population every 20 minutes or so. And scientists have been studying it for decades, so we know its genetics inside and out. All right, so we've met the tools, we've got the delivery truck, and we've found our factory. Now it's time to head to the assembly line and see how all of these pieces come together to make the magic happen. So here's how it all comes together in 5 key steps. Step 1 is preparation. You use your molecular scissors, the restriction enzymes, to cut out your gene and to cut open your vector. Step 2, ligation. You grab your molecular glue, that's DNA ligase, and you paste your gene into the vector. Step 3 is transformation. This is where you introduce this brand new recombinant DNA into your host bacteria. Step 4 is host growth. You just spread the bacteria on a petri dish and let them multiply like crazy. And finally, step 5, selection. You have to identify and pick out only the bacteria that actually contain your gene. But after you've done all of that, there's one last, and I mean absolutely critical, question you have to ask. How do you actually know it worked? How can you be sure you cloned the right piece of DNA? You have to verify everything. I mean, think about it. Your petri dish could be covered with millions of bacterial colonies. Some of them won't have the vector at all. Some will have the vector, but it'll be empty. And then, hopefully, some of them will have the vector with your gene of interest inside. You need a reliable way to find the needles in the haystack. So how do they check? Well, scientists have a few really clever methods. First, they can use restriction digestion again. They basically use the same molecular scissors to cut the gene back out and check to see if it's the right size. Second, they can use PCR confirmation, which is a technique that's kind of like a search function that finds and rapidly copies only your specific gene, proving it's actually there. And third, for the ultimate final confirmation, they turn to sequencing. And really, this quote from our source material just says it all. Sequencing is the ultimate tool. Why? Because you're no longer just looking at shadows or sizes. You're literally reading the exact order of the A's, T's, C's, and G's. It gives you final, undeniable proof. It confirms not just that you have the right gene, but that its code is 100% correct. There's just no ambiguity. So there you have it. That's molecular cloning in a nutshell. It is an incredibly powerful technique that lets us isolate and copy pretty much any piece of DNA from any organism on Earth. It's the fundamental tool that really opened the door to everything from creating life-saving medicines like insulin to developing the crops we eat. But the ability to copy is just the first step. The bigger question, the one we're left with, is what do we choose to build with these amazing tools?